Alright, welcome to our review about everything on evolution. The first part of our video has to do with common ancestry and answering the question of where and what humanity has come from. So, this question can basically be addressed when focusing on the change of prokaryotic cells evolving into eukaryotic cells. This change in cell structure brought the arrival of our common ancestors, plants, fungi, and animals. In order to understand this ancestry better, we must look at the process of how the prokaryotic cell evolved. Here, in our first picture, the prokaryotic cell only has DNA and a cell wall. In our second and third step, the cell wall is lost, the cell membrane begins its infolding process, and the cell begins developing a cytoskeleton and ribosomes. In our fourth and fifth step, the nucleus in the cell begins to form. The previously mentioned cytoskeleton has created a microtubule, and the cell begins its process to integrate a mitochondria into its system. Through the process of endosymbiosis, the mitochondria is able to be made in the cell with a proteobacterium. Voila! The mitochondria is now in the cell. Finally, the last step in the evolution of this cell is the preparation to integrate a chloroplast into the cell. The chloroplast is integrated and made with another process of endosymbiosis, this time with cyanobacteria. Now, the eukaryotic cell is complete, with the microtubule finally becoming a flagellum. Our next section in our presentation is Unit 2, Continuing Evolution. So, evolution is constantly occurring across the world. It never ends, and this is clear today with the MRSA example. MRSA is a type of bacteria that is extremely adaptable to becoming immune to antibiotics. Seeing our example, a body is completely clustered with normal bacteria and MRSA bacteria. When antibiotics are used, all of the normal bacteria die, except for the MRSA bacteria. Surviving, the MRSA begins to multiply, creating a huge amount of bacteria that is now immune to antibiotics, showcasing its constant evolution. Our next section in our presentation is Unit 3, Phylogeny. Phylogeny is the study of evolutionary development or diversification, and phylogeny is used for mapping out diseases, animals, or other examples of evolutions as well. In order to have a successful phylogeny map, you need to use the phylogenetic trees. At its base structure, the phylogenetic tree looks like a tournament bracket you would see in the month of maybe March Madness. Nevertheless, at each end of the bracket, a species we would be trying to map out would be placed there. This is specifically called a taxon. A species grouped together in the same bracket is referred to as a clade. The lines leading away from the clades and taxons are called branches. The area where two branches meet is referred to as a node, representing the common ancestor of the two clades branches. Finally, the root, located at the end of the bracket, represents the common ancestor for all taxons and clades in the phylogenetic tree. Thus, it is important to remember that a phylogenetic tree gets closer to our ancestors on the left and the present day on the right. Our next unit in our presentation is Unit 4, Speciation and Selection. The definition of speciation is the formation of a new and distinct species in the course of evolution. The first type of speciation is allopatric. The start of this begins with a single population. However, sometimes a barrier may form, splitting the population in half. Through time, the isolation has made the old same population into two different species. Eventually, these species meet back together and might possibly mate. The second type of speciation is parapatric. Starting the same way with one main population, there is another nearby population with a different niche than the main population, which remains isolated. However, and eventually, these species might meet back together and possibly mate. The third type of speciation is parapatric. This time, an already isolated population with a different niche than the main population integrates themselves back into the original main population, after stumbling upon them. Eventually, these species meet back together and might possibly mate. The last type of speciation is sympatric. Originally, with one population species, the process of genetic polymorphism creates another species which within the original population, even without a niche. Eventually, these species may mate together. Moving on to selection, its definition explains that it is the process in which the environment determines how organisms thrive. We can show selection through mouse fur examples, using a diagram that resembles a normal curve. Frequency increases as the graph goes up, while fur color gets darker the further right it approaches. The first type of selection is directional. It is when the evolved population shifts in a direction away from the original population as a whole. An example of this using the mice example is mice all getting darker fur than the original population. The second type of selection is diversifying. When the evolved population goes through an extreme shift left and right from the original population, this is diversifying selection. An example of this is mice getting super light and super dark fur than the original population. The last type of selection is stabilizing. This is when the evolved population becomes much less extreme and less diverse than the original population was. An example of this is how all mice can have average, medium, and darkish bright fur than the original population. Our next unit with the silly dead face is Unit 5, Extinction. Today, we'll be discussing the major biological extinctions that have made the way for humanity to take control. The Paleozoic era was the wet precursor to the dinosaurs, officially ending in extinction 251 million years ago. The gap between the Paleozoic era and humanity was the Mesozoic era, the time of the dinosaurs. This period is split up into 3 periods, all of which ended in extinction. First was the Triassic period, ending 200 million years ago. Second was the Jurassic period, ending 146 million years ago. Finally, there was the Cretaceous period, ending 65 million years ago, probably due to an asteroid. The final period that has led up to this date, giving a rise to both humanity and mammals, was the Cenozoic era. Our second to last unit will be on population variation, as the colorful title indicates. In order to explain population variation, we can use the bean example. The large containers are environment, the red beans are the dominant allele species, and the blue beans are the recessive allele species. Together, the red and blue beans make a total mixed and even population. So, then how exactly can we use these scenarios to show population variation? That's what you're thinking, right? Well, there is a way actually. Population variation happens when environments get destroyed. So let's say in our scenario that our jar gets destroyed. These environments can get destroyed in many ways, including storms, fires, and floods. With a natural disaster destroying an environment, or breaking the jar in our case, the population size is greatly reduced and is now clearly imbalanced in allele frequency, creating dominant and recessive alleles in the population. The final result of population variation is the reflection that the resulting population is dominant in a clear way over another. This is basically the bottleneck event. Finally, in our last unit, Unit 7, we will discuss the origin of life on Earth. The best way to map out life and growth on Earth is using a 100-yard football field example. The past is in the left end zone and the present is in the right end zone. The whole length of the field is the lifespan of Earth, 4.5 billion years ago. Life officially began on Earth at 3.8 billion years ago, or at the 15-yard line. Cells achieved the ability of photosynthesis at 3.5 billion years ago, or at the 20-yard line. Oxygen arrived on Earth at the 50-yard line, or around 2.3 billion years ago. Multicellular organisms only arrived 0.8 billion years ago, or just past the 20-yard line on the right side. The ozone layer arrived at the 13-yard line, or around 0.6 billion years ago. Just next to the 10-yard line, or around 0.53 billion years ago, animals began to develop and spread like crazy thanks to the ozone layer. Finally, humans arrived on Earth, inches away from the end zone on the right side, 0.002 billion years ago. This video was brought to you by Ozan and Seth.